The broadcast is now starting. All attendees are in listen-only mode.
Hello everyone, I'm Sally Kochmar, AGT's Education Director, and it's my pleasure to welcome you to AGT's webinar series. For those of you who are not AGT members, please consider joining the organization so that we can continue to bring you these webinars and be the voice of the genetic technologist in the community. Please also consider joining because you'll get a discount for the annual meeting. And this year's meeting is going to be in St. Louis, and it should be very different than you've experienced in the past. We've greatly expanded the number of platform presentations because we'd like to hear from you in your laboratory, your interesting cases and diagnostic dilemmas, and we've brought some other new features in that we think are going to be exciting, and we'd like to see as many of you as possible attend. And hopefully your organization will pay for your trip to the meeting if you're going to give a presentation. So if you want to give either a poster or a platform presentation, maybe it will help you get your trip funded. So please consider joining AGT. Quick housekeeping notes before we start the presentation. Feel free to ask questions during the session. In the upper right side of your computer screen, you'll see a control panel. In the lower portion of the panel, you can type in a question or comment and submit it to the webinar organizers. And you can do that anytime during the presentation. And at the end of the presentation, we'll reserve about 10 minutes for questions and answers, and I'll read the questions that you've submitted to our presenter. If you're participating as a group, please designate one person in the group to complete the online evaluation. In order for everyone to receive their certificates of attendance, that person will need to enter the names and email addresses of each person who's participating at your site today. And as a backup measure, you can send a sign-in form and fax it to AGT as well. And certificates will be emailed to the participants approximately 30 days following the webinar. Today, Dr. Heather Mason-Suarez will discuss putting a targeted gene panel on the exome backbone, the future of molecular diagnostics. Dr. Heather Mason-Suarez is an Assistant Director at Partners Personalized Medicine Laboratory for Molecular Medicine and the Cytogenetics Laboratory at Brigham and Women's Hospital, Center for Advanced Molecular Diagnostics. She also oversees training in the Harvard Medical School ABMGG Clinical Molecular Genetics Program. Dr. Mason-Suarez received her PhD in genetics from Case Western Reserve University in 2010. She completed her clinical molecular genetics and cytogenetics training at Emory School of Medicine in 2014. Her current research and clinical interests concern the application of molecular diagnosis using next-generation sequencing and microarray technologies in prenatal cases, RASopathy, and cardiomyopathy cases. Dr. Mason-Suarez.
Thank you very much for inviting me today to come speak about a new assay that we've recently launched and kind of the cons and pros associated with that assay and where we see it going in the future. I have one disclosure to disclose today. While I work at a nonprofit organization, I do offer CLIA fee-for-service molecular genetic testing. Today I'm going to be talking about two fairly common sequencing assays that are used in the clinical genetics laboratory, Sanger sequencing and next-generation sequencing. Sanger sequencing is still the gold standard for variant identification and is still used for gene panels, specifically for disorders that have a very specific phenotype and only a few genes associated with that disorder. An example of this would be maple syrup urine disease, in which it is a very specific phenotype, but only a few genes that are relatively small that are associated with this disorder. And therefore, a Sanger panel is really optimal due to pricing and completeness of the coverage for that disorder. The other type of assay used is next-generation sequencing, and there are three different ways that this is used in the clinical, specifically germline testing. We either do next-generation sequencing panels, exome, or genome. For the panels, again, we're using this for really well-defined clinical cases in which there is a fairly specific phenotype that's associated with anywhere from 10 to 200 genes. We're able to get pretty high coverage, and by that mean that the density of sequencing is fairly high. We typically in our laboratory do 200 to 400x. And this means that at every single base, it's covered within 200 to 400 different reads. So that specific nucleotide has been read on average, mind you, across all of the gene sequence 200 to 400 times. So we have fairly high coverage. We also, with this test, offer Sanger fill-in for any gaps that may be in the coverage from the next generation sequencing. So the test is complete. Unlike NextGen and Sanger, for Exome and Genome, we do not offer Sanger fill-in, and so there are gaps in the coverage. This causes these assays to be considered more of a screening assay since there are holes or gaps in the coverage. For Exome, we're really only looking at the coding regions plus or minus 15 base pairs into the intron. We cover about 22,000 genes by sequencing, but realistically, those that are associated with disease are more along the lines of less than 5,000 genes. For the genome, we are covering all genes and intronic regions, and this includes both promoters, introns, and 3' and 5' UTR. Because we don't fill in using Sanger, there is gaps, and therefore is considered a screening assay. And this assay is really best for those patients that have very complex or overlapping phenotypes in which it's very hard to diagnose, or have already undergone a very long diagnostic odyssey, meaning that they've seen multiple specialists, they've had multiple different tests, and have come back negative for any theoretically genetic causes that might be causing this. So briefly, I'd like to discuss the process of next generation sequencing, mainly to point out regions or parts of the process in which we introduce biases that cause these gaps in sequencing. For next generation sequencing, we first must extract DNA. We must break it up into small fragments, and we have to add on adapters that we ligate onto these small fragments, and the adapters are specific to which downstream platform we're going to use for next generation sequencing. These three steps are the same if you're doing genome, exome, or panel. However, for exome and panel, we also have to capture the regions of interest from the entire genome so that we're only sequencing these regions of interest. There are two common methods to do this. We can either use a hybridization method that uses probes or an amplification method that uses PCR. And here I'm showing you the hybridization method. For this, we have probes in this specific diagram here that are biotin labeled. The probes are specific to the regions of interest, so either the genes on the panel or the genes across the exome. We hybridize these to the genome, or the genomic DNA, and this hybridization step really introduces a lot of these biases. So the hybridization step is not 100% efficient, and it gets less efficient the higher or lower the GC content is. So at very high GC content or very low GC content of your region of interest can really determine the efficiency of the hybridization. After they hybridize, we use strep-avidin beads to then pull out the regions of interest from the rest of the genomic DNA. We elute the beads, and now we're left with our library. From this point, we go on to sequence the library, and I'll really be looking more at Illumina because that's the method of choice for most clinical laboratories, although there are multiple different machines out there that you can use for next generation sequencing. So for the Illumina sequencing, we separate individual strands. This can be done either on beads, chips, or spots on a slide. This then gets amplified in parallel, and we capture the amplification products locally, and this is known as sequence by synthesis. So just kind of going into more specifics here. In this figure here, we have the adapters we've added on to our DNA. These adapters allow the DNA to be captured by the slide because there's already a lawn of the sequence that binds to these adapters attached to the slides. We then use bridge amplification to amplify these so that you have a double-stranded DNA strand. It gets denatured and the DNA gets clustered in these clusters, and now it's ready for sequencing. The sequencing occurs by using reverse terminator sequencing, in which after each base, the machine is taking an image to see which base is added on to these reads. And after the reads are produced, we map them back to the genome using bioinformatic pipelines. Really, during this process of sequencing, again, there is some bias in how well things are sequenced, although less biases are introduced than by the hybridization method or the capture method. It is known that if the GC content is greater than 80% or less than 20%, next generation sequencing, the efficiency of sequencing using next generation sequencing is lower than if the content was above 20 or lower than 80%. So again, this is another place in the sequencing in which these biases are introduced and resulting in these gaps in the coverage that we see in next generation sequencing. So just to summarize the three different types of next generation sequencing, for the panels, we're looking at tens to hundreds of genes. We typically get very good coverage because our libraries are small. We're able to put more probes to each region of interest, and we're able to do a higher density of coverage, so that 200 to 400x coverage, again meaning that we are sequencing each base on average of 200 to 400 times. But of course, that is an average. So there will be regions in which that's much lower or much higher. We typically sequence exons and 15 base pairs into the intron, and of course we need the capture. And we make sure it's complete by Sanger filling in any region that doesn't have 15 to 20x coverage. For the exome, we typically cover about 1% of the genome, so these are the coding regions of the genome, and about 15 base pairs into the intron. We typically get about 97% completeness. Again, there's not as many probes per region of interest, and our coverage tends to be lower. So on average, in our laboratory, we do about 120x coverage. We do require the capture, which introduces biases or those gaps, and we do not Sanger fill them in, which is pretty typical across all of the clinical laboratories. We do offer a clinical genome. Our coverage tends to be about 95% of the genome. It includes introns, promoters, 5' and 3' UTR regions. However, I will mention that the way we interpret the genome and the exome are the same. So while we may cover more of the genome, we are unlikely to be interpreting deep intronic promoters or 3 or 5' UTR regions unless it's known that there are medically relevant variants in those regions. If we were to interpret all of them, we would end up with a lot of variants of uncertain significance, which would be very difficult to relay back to the physicians. Because we don't have that capture, however, we do get more complete coverage across the genome and more uniform coverage. We're not introducing as many biases, and therefore we can drop the amount of coverage we need to do. So for the genome, we typically do 20x coverage, and that is, we sequence each base pair about 20 times. And again, we don't offer Sanger fill-in. Mainly the reason for not offering Sanger fill-in for both the exome and the genome is it would just be very cost prohibitive. There's a lot of regions that would need to be filled in, and the lab could not support or sustain the cost of all that Sanger fill-in. For the rest of the talk, I'm really going to be focusing on panels and exomes, and I've mentioned a lot about this complete coverage of the panels because we're doing the Sanger fill-in. But what exactly does that mean? So when we look at the tests that we offer, we looked at the medical exome, and we typically get actually about 99% coverage of all medically relevant genes. So in this case, we're really looking at those 4,000 to 5,000 genes that are associated with Mendelian disorders. And then looking at our gene panels, so the following four bar graphs here are different panels that we offer in which we get coverage that's much closer to 100%, although the number of genes on these panels is anywhere from 12 up to 120, so a much smaller number of genes. We decided to look a little bit deeper and look at our cardiomyopathy panel. So at the time we had 51 genes on that panel, and with the gene panel we got very good coverage where less than 1% of the exons were not fully covered. This again is partially because we have more probes per region of interest and also because we're sequencing at a higher depth. For the exome, however, we got 15% of exons that are not fully covered. So if we were to use the exome to look at cardiomyopathy genes, 15% more would not be covered and we might miss something using the exome. When we are Sanger confirming, we Sanger confirm any region that fails. So these are larger regions that have less than 20x coverage. If there is a base in a region that is covered, but that base for some reason has less than 20x coverage, we Sanger fill in that base, and then anything that goes on the report, so any variants that we identify by next generation sequencing, we Sanger confirm. And on average, we have about 27 regions that we have to Sanger confirm per patient. And this is from our panel testing. So if we were to increase this to exome, this would increase substantially. In addition to the sequencing not fully covering the regions of interest, we also don't fully analyze every variant we identify by exome, which we do for the panels. So for the panels, we typically identify anywhere from 10 to 40 variants in our genes of interest. Many of these variants are benign and have been seen previously and previously assessed. For the exome, on average, we find 40,000 variants per individual. That is a lot of variation per person to then go in and do a full analysis on. So typically what happens for an exome is that many of these variants get filtered out based on the patient's phenotype. So here we start with 40,000 variants, and we have filters based upon what their phenotype is. So variants that are in genes that are not associated with those phenotypes will get filtered out. We do in our pipeline have a way to grab back any variants that are identified in ClinVar as being potentially disease associated or are in HGMD, which is a human mutation database. That database goes through and annotates all of the publications looking for variants that are medically relevant, and so if that variant is in that database, it again will be triggered to be caught and put back into our overall variant number for analysis. So after doing the indication-based filter, we typically can get it down to about 300 to 800 variants that we need to assess. From there, we exclude based off of the minor allele fraction. Has this variant been seen in the general population and at what percentage? If the percentage is higher than what's expected for the disease and the prevalence, we can exclude it. So our bioinformatic pipeline excludes anything in a dominant disorder that's over 1%, and then as we're analyzing, we can exclude other variants that are a very high percentage that are higher than would be expected for the disease, although that's more of a manual process. We also exclude variants that we've already previously assessed and are known to either not be causative of the disorder of our patient or have a benign interpretation, which typically leaves us with anywhere from 30 to 50 variants that are new to our laboratory that we have not seen previously. These variants are then have a full assessment. So as you can see, we really start with a large number of variants, the 40,000, and through our bioinformatic pipeline, we can bring it down to about 30 or 40 variants in which we need to go ahead and read all of the published literature on, look at in-house databases and external databases, look at any segregation studies that were done. We go to Exact and other general population frequency databases to see how often it's been seen. We look at amino acid conservation, we look at prediction tools such as polyphens, and splicing tools if it's at or near a splice site. So that's still a large number of variants for the exome, but as you can see, a lot of variants were filtered out, and so not everything we identify by the exome really gets this full variant classification. For our panels, however, all of the variants that we identify go through this process, and all of them get put into a category as designated by the ACMG, or the American College of Medical Genetics: benign, likely benign, variant of uncertain significance, likely pathogenic or pathogenic, and then go on to our clinical report. Currently, we are not reporting benign variants. However, a physician, if interested, could ask us for a list of the benign variants seen in that patient, and that's pretty typical across clinical laboratories. At this point, if we think family testing could help us reassess this variant, we will go ahead and ask family members to be tested, and depending on how helpful we think it is, we often offer testing for free to family members so we can reclassify variants and give individuals a more complete clinical report. So what are the pros of the exome? Well, one of the biggest pros of the exome is the flexible content. We're already looking at all of the genes, and so you can easily change the genes that you're analyzing through the pipelines. It's fairly straightforward. With a panel, we're making libraries that have specific genes on them. So for us to add a gene, we actually need to redesign the library and revalidate it clinically. As many of you are aware, there are multiple genes each year that are getting associated with new Mendelian and complex disorders. And often by the time we validate and launch a next generation sequencing panel, it's already out of date. This greatly can upset genetic counselors and physicians who really want these up-to-date genes to be part of the testing process. And so that can be hard to offer to them because it's so expensive to keep updating these panels. So that is one of the big benefits of an exome, is that you can continuously change what you analyze and add in more genes to each phenotype. In addition, if the correct IRBs and consents are signed, you can also use that exome data for follow-up research studies if the clinical exome is negative. In addition, the exome can give incidental findings, and there are pros and cons to incidental findings. Currently, I think the general feeling is that it is less desirable to have all the secondary or incidental findings. Many insurers have anxiety over these secondary findings. They are afraid they're going to lead to more follow-up and increased medical costs. Physicians are really unprepared to handle secondary findings and really need more education to be able to properly refer patients, to give them some type of genetic counseling, to really tell them what this means. In addition, some patients are concerned about discrimination. As many of you know, President Bush back in 2008 signed the Genetic Information Non-Discrimination Act, or GINA. However, GINA doesn't cover everything. So some things that are not covered by GINA are life and long-term disability insurance. If a patient is found to have a variant in one of the BRCA genes, it is likely that they will be denied life and long-term disability, and this law does not protect them from that. In addition, the armed forces are exempt from many portions of GINA, and so there are tighter mandates coming down for this, but they can still discriminate against genetic information for people enrolled in the armed forces. And so that's obviously a concern. However, there's a lot of research currently going on about secondary findings and how it can improve medical care and patient outcome. And so you'll continue to see in the next few years many more publications on secondary findings and how beneficiary they can be to the individual. We currently had a patient who was found to have a BRCA variant. It was pathogenic. They did not have a family history of, or at least a strong enough family history of breast cancer to have ever undergone testing for this variant. And when they went to be followed up, it was found that they did have a malignant mass in their breast and were able to get that removed at a much earlier rate than would have if they have gone through the normal process. And so you will see more things of this nature coming out, and it will be interesting to see if the perception by insurers, physicians, and patients really change to really embrace secondary findings. So lastly, next generation panels are much faster turnaround time than exomes. They can take weeks, where exomes often take months. There's a lot more interpretation in exomes. It takes quite a bit longer. And also the cost associated with panels is typically much lower than that of exomes, which can make people prefer panels to exomes. However, this is not always the case overall. So looking at this clinical case, here we have a 63-year-old female that presents to cardiology with a hypertrophic cardiomyopathy. The cardiologist orders the HCM panel. The results come back negative, and the cost is $3,600. Later on, they go on to have an exome. The result is a pathogenic variant in the PTPN11 gene, which causes a RASopathy, most notably Noonan syndrome. The cost for this test was $6,500. So overall, their cost is over $10,000 for their testing. And there are RASopathy panels out there that are cheaper. However, some RASopathies present with very mild features that are so subtle they're not always picked up. When this patient went to genetics, it was found that they had very mild features, facial features of a RASopathy. They were a little bit more noted in some childhood pictures that she had brought. However, the physician had missed that and had ordered the wrong panel, causing the test cost to be much more expensive. And this isn't an isolated one-time event. It can be very hard to actually clinically diagnose these patients, especially those that are on the mild spectrum. In the past year or two, we have put the PTPN11 gene on our HCM panel because of the difficulty in diagnosing this, and we've got 5 other patients that were seen as having only isolated hypertrophic cardiomyopathy that actually have Noonan syndrome. And when they went back to look at the patients, they did have these very mild features of a RASopathy. So we all know clinical diagnosis can be very difficult, and if they have the incorrect clinical diagnosis, the cost of testing can be quite more expensive if they start with a panel versus an exome. So just looking at what you would get at one of our panels, so this is the RASopathy panel that I had mentioned, if you had ordered it off of the panel versus the exome, you will see here that we have 100% coverage for all of the genes on the RASopathy panel on the gene panel, which would be expected because we Sanger fill in. However, these genes are pretty easy to sequence and fairly small, so most of the time we don't need any Sanger fill-in for this assay. When we had the same patient who had a negative RASopathy panel order an exome, you can look to see that most of the genes on the RASopathy panel also had 100% coverage on the exome, except for 3 genes, BRAF, CRAF, and MAP2K2, which were very close to 100%, but there was some small gaps. However, we now have all these other genes we can look at in the exome if the panel is negative. And as I mentioned earlier, often once we get a panel clinically validated and operational, it's often out of date. And within the last couple of years, we've already seen multiple new RASopathy genes be associated to the disorder. I have at least 6 listed there. And so these genes are not on our panel. And if there was a variant in one of these genes in a patient with RASopathy, we would not detect it if they only ordered the panel. So that is one limitation to keeping things on the panel. However, if you look at the turnaround time, it's 1/4 that of the exome. And the cost is also greatly reduced for the panel versus the exome. So in this case, while it might be of interest to put it on an exome because of the genes are easy to sequence by next-generation sequencing, the cost of doing and running an exome still greatly outweighs the cost of the panels. So it's really down to the cost for this disorder. For the next disease that I'm showing is pulmonary disease. So there are multiple genes on this panel, about 60, and by the panel we offer Sanger fill-in, so they're 100% complete. If we ran this on the exome and looked to see at the completeness, you would see that 54% of them are complete. However, there are numerous genes that are less than that, and a couple that are really down to 50%. So if you had a variant in SFTPA1, you would likely miss it by ordering an exome, and this gene might be of great interest to the clinician and the patient if that's where their variant is. The turnaround times for this panel, because the genes are a little bit more complex, is longer, and the cost is longer because the cost is higher because the Sanger follow-up is much greater. And so the cost and the time frame is a little bit more similar to the exome, but still we wouldn't get all the genes that we want to be fully covered. So looking just to summarize the two spectrums of these tests, for panels we get complete coverage, which can be very important to the physicians and to the patients, and all variants are reported and thoroughly assessed. For the exome, however, we have much more flexibility in what we can identify and new genes that we can analyze. We can identify secondary findings, which might be of interest to patients and their physicians. And there is a potential with the proper IRB and consent to use any exome data that's negative to do gene discovery or identify the patient's cause of disease. So we wanted to know, is there a way that we can combine these two assays to give patients and physicians the best of both worlds? So what we came up with was an exome-based gene panel, and we wanted to start by offering whole exome sequencing, but then ensuring that we covered the regions of interest or the genes that are highly associated with the disease at 100%. So there was no way we could miss a variant that could be causing disease. We would do a full interpretation of all the variants identified in these genes and report them back. But if the panel was negative or inconclusive, we could now analyze the entire exome. We wouldn't be able to offer Sanger fill-in because that would still be too expensive, and we would offer reports for variants that are likely path-pass or a variant of uncertain significance that's highly associated, in a gene highly associated with disease. So how are we going to set out to do that? First, part of our development was really we needed to pick a gene of or disease of interest. And that sounds fairly straightforward, but there were a lot of different things that we had to consider. First, to pull off this test, it would still likely be as expensive as an exome. So the cost would be about $6,500, which is really expensive. So we couldn't take our rhapsopathy panel and put it onto an exome because right now we offer the rhapsopathy panel at $1,500 and nobody's going to purchase it at $6,500. So some of the things to consider were what was already available. Was there any tests that really were of interest to physicians and patients but not yet put on a gene panel so that we weren't competing with panels that were already a fraction of the cost? And what was the total cost of the tests that were available? Were there some tests available but they didn't have enough genes and the costs were already pretty high? And in addition, to be completely comprehensive, would we have to add any additional assays? Next generation sequencing can detect copy number variants, but it's still copy number variant detection off of it is not very good. There's still gaps in our knowledge there and our ability to use that. To be able to detect repeat expansions is very difficult. Pseudogene analysis can really only be done using long-range PCR, and then methylation detection, if that was involved in the comprehensive testing, we would have to offer other tests to kind of supplement it, and how would we consider that in the overall cost and what physicians, insurers, and patients are willing to pay for. So we were able to come up with a disease that kind of met our criteria of not having other panels offered yet, so we could justify the high cost of the exome. Next, we needed to identify what genes we were going to test for that disease. This sounds, again, like it should be a very easy thing to do, but if you look in the genetic testing research or genetic testing resource, you'll see for any different disorder, the panels, what genes they offer for those panels can be very different depending on which laboratory you're sending your testing to. So here it's just looking at cardiomyopathy testing. You can see that most laboratories offer the sarcomere genes, and those are pretty well known to be associated and really need to be on the panel for a comprehensive test. However, some laboratories offer all of the storage cardiomyopathy genes and some do not. As you get out to the syndromic, such as your RASopathy, the number of laboratories that offer that are even less. And then in addition, there are other genes, almost 100 other genes that differ between laboratories that some laboratories feel should be a part of the test and some laboratories feel shouldn't be a part of the test. And so really curating your gene content can be very difficult. We worked with some clinicians and determined 278 genes of interest that were determined to be candidate genes for our disease area. So we wanted to look at these genes and see how well they were covered in our past exomes. And so we did a preliminary analysis looking at how much potential Sanger fill-in we would have to do to offer all of these genes on our gene panel. So we looked at all 278 genes to see how well they were covered by the exome and how many follow-up regions we would have to Sanger fill in. And when we looked at this, you can see here that there were about 120 different regions that we would potentially have to Sanger fill in for any given patient. Our typical panels that we have in the laboratory, we often do 27 or less amplicons per sample. And so going up to 200 or 120 is just really not feasible for our operations and keeping this within a cost that would be acceptable to insurers and physicians. So we had to go back and really curate these genes some more to determine what we really needed to be on our panel. So the way we did this was we used ClinGen, which is an entity funded by the NIH that is trying to kind of uniform classifications of variants and also genes. And we used their criteria for defining if our genes of interest were truly associated with the disorder. So we looked at all 278 genes. This was completed by a postdoc and senior technician in our laboratory to determine this. And we found that 20 genes really had very little evidence, if any evidence at all, for a causal role in disease. So these are genes that often the physicians thought were of interest, and when we really interrogated them, we found very little evidence. There were 50 of these genes that had limited evidence, so limited meaning less than 3 families that had a possible pathogenic variant. Or it could mean multiple variants without sufficient evidence for pathogenicity. So these multiple variants in one gene, but none of the variants actually had enough to be really considered pathogenic on its own. 50 genes had moderate evidence, So in this case, there were greater than 3 families that had possible pathogenic variants and some supporting functional data. There were 63 that were strongly associated, meaning that multiple families had pathogenic variants, there was supporting data, and there were at least 2 independent published reports by 2 independent research groups. 95 genes had a definite role, so these were genes that were found to be repeatedly demonstrated, had multiple pathogenic variants, and upheld for at least 3 years in publications. So that's over 3 years of every laboratory looking at these genes saying, yes, they are associated with this disorder. So what we decided to do was then take the definitive and strong genes and determine how many amplicons we would need to fill in for that if we were to run this disorder on the exome backbone. And we were able to greatly decrease it down to close to 60 amplicons. However, this was still too high to be feasible for our lab to do this many amplicons, keeping it within this price range and within a decent turnaround time. So again, we had to go back to the drawing board and really further curate these genes and determine the test content. And what we ended up deciding to do was to create subpanels. So we took genes of interest, looked for specific phenotypes, and created these subpanels that had minimal overlap so that the physician can order a subpanel and then reflex to exome if negative. So when we then looked at these subpanels, which were anywhere from about a little over 40 to 60 genes, we were then able to get the amplicon follow-up down to a reasonable number, close to 20 amplicons per panel. So once we were done with that, we designed the primers for Sanger follow-up, we validated, and we clinically launched this assay. So just to follow up what our workflow is currently for this assay is the patient comes in with a phenotype and the physician can order one of 3 panels depending on kind of the sub-phenotype of the patient. We offer whole exome sequencing, and then we offer 100% coverage for those 40 to 60 genes where all bases have greater than 15x coverage by the next generation sequencing, and those that don't are filled in by Sanger sequencing. We offer a full interpretation for all of those variants, and we do a report that includes likely benigns, variants of uncertain significance, likely pathogenic, and pathogenic variants. If the patient's report is negative or if it's inconclusive, meaning we only found a variant of uncertain significance, we then reflex to the remaining exome. We don't offer Sanger fill-in for the rest of the exome, but we do look at the entire medical exome, and we report anything that's likely pathogenic, and also any VUSs that are in genes highly associated with the phenotype of the patient, and that we would want to do further family studies on. So in summary, our targeted panel, we have 100% coverage, and we report everything except for benign variants, although a physician can request the benign variants. For our whole exome sequencing, we do offer the indication-driven gene list. This we do not offer fill-in, and we report pathogenic, likely pathogenic, and some VUSs or variants of uncertain significance that are in genes highly associated with the disease and potentially might be causing their disease. If that's negative, we reflex to the entire medical exome, so looking at those almost 5,000 genes. Again, there's no fill-in, and we report the same thing, and we offer incidental findings that the patient can opt out of. Again, there's no fill-in for that, and we only report pathogenic or likely pathogenic variants for incidental findings. We never report any variants of uncertain significance. For our newer panel off of the exome backbone, it's kind of the best of both worlds. We offer 100% coverage for those genes of interest that are part of the subpanels. We Sanger fill in all of those genes, and we report anything from pathogenic to likely benign, with the possibility of requesting benigns if the physician was interested. We then expand. If the test is negative, we expand it to the entire medical exome. So looking at those 4,600 genes, we do not offer Sanger fill-in for those, so there will be some of those gaps that I mentioned. There is the possibility of missing things, and we report likely path, and then some VUSs. We also, for this, we offer the potential of incidental findings, so the patient has to determine, check off yes or no if they want the incidental findings, but it is offered to them. There again is no fill-in, so things can be missed, and we only report pathogenic or likely pathogenic variants. So this really gives us the best of both worlds. Of course, the big con for this is that it's expensive. We're unable to really decrease the price from that of the exome. So the cost is what we would charge for an exome. Although you are getting more, you're getting a lot of Sanger fill-in, and the turnaround time is expensive. So while the benefits of offering it this way is great, the cons are that it is very expensive, and currently it's very hard to put panels on the exome backbone because of this. However, as the exome costs decrease, we plan to see many more panels being put on an exome backbone. And little by little, we will be moving more and more of our panels to the exome backbone as we can, because we think that this is a better assay overall and allows for really the best of all of your options. So with that, I would like to take time to thank everybody that I work with at the LMM. We have a very big group of individuals that really make everything that we do there possible. A lot of the work from this was also done by Birgit and our LMM development team, Beth Duffy and Mark Bowser, and I will welcome any questions that anybody would like to ask about this or anything else that came up during our talk. So thank you.
Thank you, Dr. Mason-Suarez, for your informative presentation. To remind everybody, you can submit questions in the control panel to the right side of your screen by just typing in a question, and then we will get them to our presenter. Dr. Mason-Suarez, do you have any experience with whether insurance companies would cover the exome-based testing versus the panel?
So currently, the way that we are actually coding it, and even though the costs are more expensive, we're coding it as a panel. So although I haven't looked into how our reimbursement has gone for that, we expect that it'll be reimbursed more like a panel than an exome. So when exomes first were launched, it was very common for exomes to actually get pretty high reimbursements until the insurance company figured out exactly what they were and kind of put a stop to that, and then really wanted people to order panels. So we've tried to charge it more as a panel and use a CPT code as a panel. So I believe currently they're getting reimbursed in this order.
Great, thank you. At this point, we don't have any other questions that were submitted. Thank you everyone for participating in the webinar. Again, you will receive an email from AGT that includes a link to an evaluation, and please take a few minutes to access it and provide your feedback on today's sessions and give us a heads up on any future webinars that you would like to see. The webinar today was recorded and it will be posted on the AGT website very shortly. And the quiz for last month's webinar should be posted by the end of the week for any of your colleagues who did not get to view that live. It will be available on the AGT website, as will this presentation. We hope you'll join us for upcoming webinars. We do have one in December on the 15th of December at 1 p.m. Eastern, and that is Dr. Alexis Carter talking about next-gen sequencing informatics. So we hope you can all join us for that one, and keep an eye out for the registration email that will be coming. And again, we hope to see you at the annual meeting in St. Louis, Missouri, which is scheduled for June 15th to 17th next year. Thank you very much.